Hello friends and welcome to the my channel Easy Life Science. Here in this video lecture I am talking about a capsule staining by a manual method. So please share my videos and press the bell icon and subscribe my channel Easy Life Science. Let's start. Here in this image there is a one bacterial cell and it has a capsule around their cell wall. Now about the capsule, some bacteria have a layer around their cell wall, and this layer is made up of a mucus or a gummy substance, and this is called as a capsule. The capsule is composed of 98% of water and 2% of polysaccharide and polypeptides. But some polysaccharides present in a capsule are different. For example, in a Bacillus anthracis, the capsule of it is made up of a poly-D-glutamic acid. The capsules are clearly visible in a light microscope when a negative stains or a pressure stains are employed. What does this capsule do? These are some significance listed here. The first, the capsule helps the bacteria against a phagocytosis. When any bacterium or any bacterial pathogen enters into a vertebrate body, the vertebrate body tries to kill it by a defense mechanism or by a immune system, and in an immune system the phagocytic cell plays an important role. This cell firstly engulf the bacterial pathogen, and after engulfment, by synthesizing a lysozyme like enzyme, they kills that bacterial pathogen. But because of the presence of a capsule around some bacteria, these bacteria have a resistance towards such phagocytosis. The capsule protects against a desiccation. The capsule is made up of 98% of water. During the growth of organism, when there is a lack of water or when there is a desiccation, at this time the bacteria make a use of water from that capsule. The capsule provides a food at time of lack of nutrients in a medium. Because the capsule is made up of a polysaccharide and a polypeptide, these are a food, and at time of lack of nutrient during the growth, bacteria make a use of that food in form of polypeptides and polysaccharides present in a capsule. The capsule is a virulent agent. It increases a pathogenicity of the microorganisms or a bacteria. For example, we know that the Streptococcus pneumoniae, this organism is a capsulated organism, and capsule antigens are present in the capsule. It causes a pneumonia, and because of the presence of a capsule, this organism is more virulent. The capsule prevents from a complement mediated bacterial cell lysis. The complement mediated bacterial cell lysis means when the bacteria or bacterial pathogens enter in a vertebrate body, at this time the complements act on it, and as the organism present a capsule around their cell, it does not get identified by an immune system, so it gets protected from a cell lysis. The capsule gives protection to the anaerobic organism from oxygen toxicity. The oxygen is toxic for an anaerobic organism, and anaerobic organism or some anaerobic organism produces a capsule, and so that they get protected from oxygen toxicity. They exclude bacterial viruses and most hydrophobic toxic materials such as detergent. They also aids bacterial attachment to the surface of solid object in an aquatic environment or to the tissue surface in a plant and animal host. At time of to cause infection, the bacteria make an attachment to the surface of some tissues of plant and animal. So this is a adhesion organ for the bacteria. Some organisms are there which shows a capsule around them, and these organisms are called as capsulated organisms. Some organisms are listed here. Basically, the Streptococcus pneumoniae and Staphylococcus aureus. These two are a capsule producing gram positive pathogens. Now some about a staining. Staining, it is a process to give a color to the microorganisms by using a stains. Staining further classified as a simple staining, differential staining and spatial staining. The simple staining, it is also called as a monochrome staining or a positive staining. It is useful for a study of morphology and a study of organisms like their shapes and their arrangement. The differential staining technique, that is a gram staining and a acid fast staining. By using this staining technique, we can differentiate the two types of organism. By performing gram staining, we can differentiate a gram positive bacteria and gram negative bacteria, whereas by performing acid fast staining, we can differentiate acid fast bacteria and acid non fast bacteria. Spatial staining technique, in this staining technique, the particular organ or a particular component of the cell get colored and we observe it. Capsule staining, flagell staining, spore staining, cellular staining or nuclear material staining, these are some example of spatial staining. Now about a dye, it is a colored substance and this substance has a affinity to the substrate to which it is applied. Dyes are of two types, a basic dye or a acidic dye. Also another type is there, that is a neutral dye or a neutral stain. Now the basic dye possesses a positive charge on their chrome 4 group. Example of it: methylene blue, basic phosphine, crystal violet, sapropyrin, meritite green. These are some example of basic stains, and these stains give a color to a acidic component of the cell. The next, that is acidic dye, they possess a negatively charged chrome 4 group. Example: eosin, rose bengal, picric acid, acid phosphine, Congo red, and India ink. These are some example of acidic stains, and they gives a color to a basic component of the cell. Then the third type is a neutral stain. This shows a presence of positively charged as well as negatively charged chrome 4 groups. For example, Giemsa stain and Leishman stain. 5. Capsule staining. We know that the capsules are non-ionic. They don't show any charge on it. So neither acidic or nor basic stain will adhere to their surface, because the most capsules are water soluble and a simple stains will not adhere to them. Since the capsule is a major virulence factor in a major disease causing bacteria, so it is essential to identify by using a staining technique. Now the principle of capsule staining, the negative staining method will be employed for a capsule staining, in which a translucent or darker colored background with stained cells but unstained capsule will be observed. The background is stained with the dyes like India ink or a nigrosin or a Congo red. These three are the acidic stains, and by using any one from these three stain or such acid stain, the background is colored. And for a coloration of a cell or a cytoplasm, the counter stain will be employed. For this purpose, there is a use of crystal violet, methylene blue, or a new phosphine, or any other basic stain, and the color of that basic stain will be taken by a bacteria. Capsule will be appear colorless with stained cells against dark background. The background color, it depends upon a color of used acidic stains, whereas color of cell it depends upon used basic stain. Capsules are fragile, they are delicate, and they get diminished, desiccated, destroyed or distorted by a heating or a simple washing. So a drop of serum can be used during a smearing, and this drop of serum will enhance the size of capsule and make it more easily observed with a typical compound light microscope. Now, some principles of Manual's capsule staining. The method given by a scientist manuals for a capsule staining. This staining technique uses the principle of negative staining for a demonstration of capsule, in which background is stained, cytoplasm is stained, and capsule remains colorless. The requirements: a clean grease-free slides, nichrome wire loop, 24 hours old culture of capsulated bacteria, 1% congruent solution, and manual stain. These are the requirement for it. The next, a composition of manual stain. It has a 4 component: 1% acid phosphine, 5% phenol, 30% FeCl3, 20% acetic acid. The 1% acid phosphine, it is a basic dye and it stains the cytoplasm or a cell. The 5% phenol, it increases a penetration power of a stain, that is a acid phosphine. And FeCl3, it is a chemical fixative agent, whereas the acetic acid, it changes the color of background from red to blue. Then the procedure, the first step, that is prepare a loopful culture smear on a slide by a nichrome wire loop. After this, air dry the slide and don't heat fix it. Then put a drop of 1% congruent at one end of the slide and spread it across the smear with the help of another slide by holding a slide at a 45-degree angle. Then dry the film of a congruent. Next point, flood the smear with a manual stain and wait for 4 minutes. Next point, discard the excess of stain by a blotting paper, but do not wash with the water. Then dry the slide and observe under oil immersion objective. Observe for blue colored background, pink colored bacterial cell, and colorless capsule. Here, blue colored background, pink colored cell or a cytoplasm, and a colorless cytoplasm or colorless capsule. Another image, red colored cell, colorless capsule, and blue colored background. Here are some example of capsulated bacteria: Klebsiella pescis, Azotobacter pescis, Rhizobium, Bacillus, and Xanthomonas. So thank you for watching my video. Subscribe my channel Easy Life Science, like my video and share it to your friends, and press the bell icon so you will get the notification of newly uploaded videos. Thank you once again.